Morning and good afternoon, depending on where you're joining from. Welcome to the AgriFutures Chicken Meat Program webinars. If this is where you're expecting to be, you're in the right place. If this is not, please stay because you're in for hearing about something new and interesting. I'd like to start by with an acknowledgement to country. AgriFutures Australia acknowledges the First Nations people of Australia as the traditional custodians of the land and waters on which we live, learn and work. We pay our respects to past, present and future elders of these nations and any First Nations people on this webinar today. And specifically for me, I'd like to acknowledge the Ngunnawal people of Australia, the traditional custodians of the lands and waters where I am hosting this webinar from today. Now, I think it might be a little bit late for a Happy New Year, but welcome to the first webinar for the program for 2025. It's great to have your company. Before we get into exploring the research being featured in this webinar, a little bit of housekeeping. We will be recording today's session and that recording has started and making that available through our online channels. As I run through the housekeeping items, there will be an introductory poll that pops up onto your screen with some questions just to gauge who's in our virtual room with us, and we'd be grateful for your responses. As this is a webinar, your video and audio functionality are not enabled, and that just allows us to best utilize the Zoom screen real estate to view the shared content, and it also means that we can minimize background noises and distractions to our invited speakers. We will have time for Q&As or question and answer at the conclusion of the webinar content. In the meantime, there is the dedicated Q&A chat function for you all to ask any questions over the course of the webinar, and that's just located along the bottom of your Zoom screens. So, firstly, an introduction to myself. My name is Sarika Pandya. I am the research manager, part of the broader AgriFutures, Levied and Emerging Industries team, and I specifically look after the chicken meat research development and extension program. So this webinar today, it's the 7th in the series, and we like to host expert presenters to provide you with the latest information on topics that are relevant to industry, as well as the research priorities in the AgriFutures chicken meat RD&E plan. So today's content, it doesn't align to one single topic like many of our previous webinars. Rather, it features some of the new projects that commenced in June and July of last year. So that was in response to an open call that we had advertised, which sought proposals that were innovative and had the potential to bring transformative change in some of the priority areas within that RD&E plan that I mentioned a moment ago. The program called for proposals that addressed current needs and challenges of the industry, and at that time it included, and still does, unprecedented period of biosecurity threats, as well as the industry that is continuously striving to strengthen its sustainability credentials. As a result, the program did contract 8 new research projects, and today and it covers 3 of the 5 RD&E priorities. And this session will explore how some of the program's new projects, specifically 3, are enhancing long-term industry adoption, supporting sustainability credentials, as well as investigating disease prevalence. So I hope you enjoyed today's content. And with that, I will start to introduce our first speaker. I'll kindly ask Sam and John to turn on your screens and commence sharing your slides. So Sam Moore is a senior behavioural scientist that leads the Australian agri-environmental portfolio of the company at Evidn. Sam has worked across numerous agricultural settings to design and deliver behaviour change programs to increase the adoption of best management practices. And a core component of his role is deep qualitative engagements with agricultural landholders, and that's to design meaningful solutions based on the unique needs and challenges of each industry and community. And part of Sam's role includes regularly engaging with representatives of grazing, cropping, grains and horticultural industries, and now the chicken meat industry. We also have Dr. John Pickering, who is the Chief Behavioural Scientist at Evidn, and he also has a very long history of applying behavioural science in agriculture settings in Australia. And today they'll both be informing us today on using behavioural science to enhance extension and adoption outcomes for the Australian chicken meat industry. So with that, I've spoken quite a fair bit. I'll hand over to you, John and Sam.
Thanks so much, Sarika. And good afternoon to everybody, and thanks for joining us. We're looking forward to walking you through our research program and very grateful to being involved with AgriFutures and the group on this topic of behavioural science. in the context of extension adoption in chicken meat. And I'll dive straight in. Sam, we might go to the next slide about where kind of Evidn's coming from here. And it's this idea that in agriculture, many of the big challenges and opportunities essentially implicate the attitudes and behaviours of people. And of course, there's growers, there's processors, there's all sorts of different groups and individuals along the supply chain. And therefore, the extent to which we can understand and potentially work with people to modify various attitudes or behaviours is probably going to be key to how we solve some of the big challenges, especially when it comes to adoption and, dare I say, extension. And behavioural science provides at least one set of perhaps tools or insights that can help us with that. It's not obviously the only part of the or only component that we need to consider, but it's an important part nevertheless. And there's quite a bit of evidence to suggest why that indeed can be useful in agriculture here in Australia and elsewhere, and that Evidn has also been involved with. And so when I'm framing up and we're thinking about our program of work and Evidn's role, and we might pop to the next slide if that's okay. Essentially, the way to think about it, in the very broad sense of the term, is this idea of we do two things. And the first is this behavioural analysis component, which is essentially trying to understand why adoption in this instance is or isn't happening. And then the change part, and it's all at scale, is what we're really interested in thinking about, is what can we do? What can we implement? And in this case, for this program of work, we're thinking about what are the insights and the tools and the resources and the training that we can develop and deploy across potentially multiple other research projects that are happening within this particular sequence of work. So that's very briefly and broadly about Evidn. I think, Sam, if we go to the next slide, it gets to some of the specifics of the aims of this particular project. And the first one there being about this idea of identifying the driving and restraining forces that are present across the industry that are relevant to improving bird performance. And that's based simply on the Lewinian theory of social psychology, that behavior is an equilibrium between those driving and restraining forces. And it's often those things that are restraining behavior, or the barriers they're sometimes called, that are the most important to firstly understand and then work out how to mitigate in order to bring about a change of practice or behavior, if you will. And so we pay a lot of attention to that, and that work is currently underway. And then on the back of that, we're looking at the development and delivery of a training program for the chicken meat workforce, and a toolkit in behavioural science that, as I alluded to a moment ago, could be more broadly deployed across AgriFutures programs and other levied industries for that matter as well, but certainly chicken meat as a priority. So that's broadly what we're doing. I'll pass now to my colleague Sam, who will just give a little bit more in the way of specifics around the program, some of the steps we're taking to do that, and then we're keen to have some discussion or questions with the group as permitted. So Sam, over to you.
Awesome. Thank you, John, and thank you all joining today for the opportunity to share our work. What we're really keen to do now, as John mentioned, these aims actually really nicely map onto essentially the main phases of work we'll be going through over the coming months. Obviously, phase one well and truly underway, that analysis. We're really keen just to talk everyone through what these all look like in a little bit more detail, and then wrap up and let you know where this is all heading from here. So firstly, that analysis that John mentioned earlier is really the first activity, which is well and truly underway at the moment. Working from the very far right here, the end outcome here is really about how we can understand those driving and restraining forces and develop some behavior change strategies that we think will be really helpful for overcoming them, which will then inform the training program in due course. But to get to that point, there are a number of different inputs we're using, which you're seeing there on the left. Firstly, the literature review, understanding what we already know about human behavior and the broader system and context that's influencing this particular challenge and opportunity. Looking at the desktop review, concurrent projects, initiatives, policies, programs that are underway to get a broader read on the industry. Also understand what's been really effective in this particular context, or some learnings from previous projects as well that we can make sure we keep across. The third input is by far the most important and time and resource intensive on our end, which is really understanding and connecting with some of the key stakeholders across the supply chain and really doing so in a really deep and meaningful way to understand their perspectives, what's working really well, what are some potential challenges or barriers that we can potentially do something about as well. These take the form of individual discussions. They can be part of existing industry events and of course through virtual and online formats as well. What all this leads to, as John mentioned, is some of those driving and restraining forces, in this case to extension and adoption outcomes, primarily as it relates to bird performance, and doing so as a function of the different stakeholder groups, but also prioritizing these as well based on their impact and their feasibility for behavior change, which then gives us a lot of precision about where and when we need to target and start to shape up the training program. So the broader thing to mention here is that this is a really good framework. There is a lot to it, but it's a really important way of understanding behavior across the whole industry, not just one particular stakeholder group, and also understanding the broader system that surrounds this issue as well. The second component is obviously the program itself, and the analysis will be really important for shaping up exactly what this looks like. The main focus here is looking at what those barriers are and what are some tools and strategies that we can design to help to mitigate or potentially overcome those as well. The real focus here is looking at the skills and capability of the existing workforce and how we can improve their relationships and extension and how the information is getting out there to hopefully then in turn improve bird performance in due course. We've called this a training program. The main outcome from our end is making sure this is as practical and feasible as possible for the industry, and it will work most importantly. So there's a lot of flexibility about what this will look like and how it's delivered. That's really what that third point there is getting at. There's opportunities for this to be embedded as part of existing events and meetings. Of course, it could be delivered through standalone opportunities if required, but the main outcome again from the analysis will be to help shape up exactly what this looks like, what the content will be, what's the delivery format and messaging, etc., that we need to consider to help to overcome some of those barriers. The final stage here is the last few months of the project, and what this is really about is making sure that any particular outputs, outcomes that we deliver can continue well beyond the length of the project. And so making sure we have a way of firstly embedding all of these tools and skills and knowledge that we'll develop back into the industry, and then potentially looking at how this could apply to other levied sectors or programs in due course as well. Basically what this will look like is a pocket guide or a toolkit that will be developed for industry stakeholders, which basically summarizes all of the different training tools and strategies that we'll be rolling out in due course. So that's a quick fly through of each of the main activities. I promise this is as technical as we get, but just wanted to give you all a quick snapshot of how this all hangs together. As we mentioned throughout, it really is that first behavioral analysis component that is so important here to shape up the outputs and outcomes of the program. But what you're seeing here on the left is the analysis really helped to inform firstly a number of different outputs. 4 you're seeing there in that kind of left middle column, what the drivers and barriers are, the development and delivery of the training program itself, looking at some monitoring and evaluation indicators to make sure we can track the program and behavior change, and then the toolkit itself to embed into the industry and potentially others. This will be designed for a number of different audiences as well. Obviously, first and foremost, the chicken meat industry and workforce, concurrent programs and projects that are operating in this space to make sure we can align and leverage and collaborate with all the great work happening across the industry, and then potentially other levied industries as well. There might be some overarching tools or strategies that we think might be beneficial in due course. Essentially, what we hope this will all lead to is improved bird performance, improved knowledge, skills, and capability of the industry staff that are involved in this program, and then some broader social and economic benefits. But really, the main focus here is how we can improve that, dare I say, soft skills of industry staff that are working more one-on-one on the ground, and in doing so, making sure we can get the best support, advice, information out to the people who need it. I guess in wrapping up, there's a few next steps we're undertaking, but really keen to have some of these more interviews and engagements with a good snapshot of the industry over the coming months as well. And we've listed just there in dot points a couple of different types of the industry we'd be really keen to meet with. Of course, industry groups and bodies, government representatives, and then some of the key staff on the ground as well, where appropriate to do so. Some of the transporters, the vets, farm service staff and managers, and of course, where appropriate, the growers themselves as well. And then this will be running through until about mid-year, where we start to roll out and design the training program and toolkit moving forwards. So thank you all very much for the opportunity to share where this is all up to. We would definitely love the opportunity to speak with those who are interested in more detail. We've included our contact details and emails here for those that are interested, but thank you all very much.
Thanks very much, John and Sam. And just a reminder to everybody here, the slides and the information shared in this webinar will be made available afterwards if you hadn't taken a screenshot of the email, those email addresses that were there on the screen. I'd also like to remind our audience that we do have the Q&A function. John and Sam, there is an interesting one there in the Q&A, but I will come back to that at the end after our following two presenters have shared their content. So now I'll move us over to the, I'll move on to the next project that is being featured. So Mary Frances Copley is a senior consultant at Integrity Ag and has a background in agriculture and economics. She has a particular interest in environmental accounting and sustainability for chicken meat, egg and pork production, and has conducted life cycle assessments, developed emission reduction pathways, and a host of other agri-environmental projects that advance sustainable outcomes for these industries. So during this next segment, we'll hear from Fran about the project titled Techno-economic Emission Reduction Pathways for the Australian Chicken Meat Industry. Thank you, Fran.
Thanks very much, Sarika. And yeah, good afternoon, everyone. Thanks for attending. I've done many of these webinars before across different industries, and yeah, I think chicken meat always gets good attendance, and hopefully it's a sign that everyone's very interested in the work and the research that AgriFutures is supporting, and further interest as John and Sam were just alluding to, in how do we sort of carry that forward to adoption and implementation on farm. So as Sarika mentioned, Integrity Ag has a project at the moment around developing techno-economic emission reduction pathways for the Australian chicken meat industry. And just before I get into, I guess, what that project entails, where we're going, and what the benefit of that will be, I'd just like to sort of set the scene a little bit, because every stakeholder we speak to has a different interpretation or different priority when it comes to sustainability or climate or nature. And so it can be quite easy to sort of get lost in the weeds a bit of, well, what are the overarching themes sort of governing that? And when it comes to chicken meat, there are sort of three main ones. In terms of emission reduction, the federal government has really set our path there, or successive federal governments, through their commitment to the Paris Climate Agreement, and ultimately what the government is pushing forward is around a 43% national emission reduction target by 2030 from 2005 levels. And sort of the long-term component of that is to then reach net zero economy-wide emissions by 2050. On the other side, we have our major customers who have, for their corporate reporting, for ESG, for ESG reporting and all that, they have their own Scope 3 sort of supply chain targets that are increasingly being set. And sort of the voluntary standard that most of those customers are aligning themselves with is the Science Based Targets Initiative. And what is required there, although it sort of generally harks back to the same principle of, keeping global warming well below 2 degrees, 2 degrees additional warming, or between 1.5 and 2 degrees, exactly what's required as part of an SBTI aligned emission reduction pathway differs slightly to what, a federal government aligned pathway would look like. And that then really, brings us into the third major component of, well, where do our farmers sit? Where do our primary producers sit in this space when, we're trying to operate sustainable and profitable businesses, and recognize that, no one's going out there wrecking the environment willy-nilly, really. Effective environmental management, effective business management should be a sort of precursor to some of that, sustainable business operation. But that being said, how do we operate in this environment given these, in some ways conflicting, in some ways supporting elements from, customers and government? So where these projects really come from is the fact that we know these stakeholders are asking for emission reduction. And, hand on heart, we know that's going to increase the cost of production for the chicken meat industry. But the question is how much? There currently is no published, published industry study that's looked at these different emission reduction targets in the context of chicken meat and set out, what's required, by when, at what adoption rate, in order to achieve these targets, so that, those conversations can be had not only around what it would take to achieve this, but also, a broader reckoning of, is this achievable in the context of agricultural production? So what's technically achievable, what's financially achievable, and how do we get there? So our objectives in this project are to model, set out industry emission reduction pathways and compare, the level of ambition, the strategies required, and the costs between those different targets. We'll also report a detailed disaggregation of the Scope 1 emissions for the industry. So for those not familiar, Scope 1 basically refers to the emissions arising directly within and because of Australian chicken meat production. The other sort of scope of emission that often goes hand in hand in that is Scope 2, but that corresponds directly to grid electricity consumption. Whereas under Scope 1, we have multiple sources. We have, emissions from manure, emissions from gas use, emissions from effluent treatment at meat processing plants. And so understanding the relative contribution of those is important to identifying the hotspots for emission reduction and analysis. We'll also conduct an economic assessment of the costs of these emission reduction pathways. And then moving forward, we'll publish those findings in the peer-reviewed literature to give this study, credibility, so that whether it be, a particularly interested party, a stakeholder, one of the vertically integrated processing companies, in conversations with others, there is a piece of evidence to refer back to around this. And as part of this process, we'll also identify, what priority topics for research and development and also extension are, for industry to in future meet these particular targets. So I won't spend a whole heap of time on our project methods, as they can get into the weeds a little bit, but essentially our emission reduction pathways will consider both absolute, so total industry emissions, and also the emission intensity reduction. So think of that as emissions per kilogram of carcass weight. And, in our identification of viable emission reduction strategies and technologies, that's, I beg your pardon, been informed by sort of a wealth of past research and literature reviews. We've done that first stage economic assessment. And then where we've gone is presented sort of these past two stages to an industry steering committee, which is made up of, representatives from the major companies and major grower networks, to really go, okay, well, based on your understanding of this, what's a reasonable time frame for implementation? Do these cost assumptions look right? Does this assumed adoption rate, align with what your expectations and understanding of your supply chain is? We then basically put forward the same assumptions to that group for anonymous feedback. Part of the reason we've done that is that when you get a group of people in the room who are from the chicken meat industry, as many of you are probably aware, there are a lot of closely held production and cost related components And so this was one way of, I guess, removing that and ensuring we can get a complete picture, but we don't need to put anyone on the spot to kind of, I guess, reveal their secrets in a meeting with others. So then we'll move forward to that journal publication, which will be our first primary output. But we'll also produce another output, which is a bit more of an administrative report that'll go to AgriFutures, but it'll also include, I guess, a detailed summary of what those recommendations are for research and development, but also extension. How do we carry, the findings of this project forward and democratize the results across all levels of the industry? And so what that means is, how do we distill the most important parts of this project for a contract grower versus a major vertically integrated processing company, because of what's of most relevance, interest, and I guess benefit to them will differ between those two stakeholders. So in terms of our timelines, we're well underway with this project. It commenced, in the middle of last year. We presented the draft emission reduction framework in December. Our draft project outputs will be completed this month, with the final outputs completed in May, and then that journal paper. It's a bit to be confirmed depending on sort of the timelines from the journal itself, but we hope to see that published in late this year, maybe November. So as mentioned, or what this kind of implies is we are fairly advanced in this project. And, whilst I would have liked to have put some results in front of you today, we are sticking to sort of our agreed project flow. So, once those results have gone to the steering committee and to AgriFutures for review, we'll start looking at disseminating them to industry via extension activities in the next few months. So what that looks like is we do have a webinar series planned for growers. And I guess the point there is that we want it to be very practical and outcome focused. So it's focused on that on-ground implementation of strategies and technologies. So how do you do it? What does it cost? What are the trade-offs? What do you need to be aware of? What does it get you in terms of emission reduction if you're so interested? And then also placing those emissions from contract grower farms in the broader picture of the industry. So what do they actually represent as an industry total? What are the vertically integrated processing companies going to be asking moving forward? Because they do have emerging requirements from the Federal Treasury around public reporting of their greenhouse gas emissions. So, what do growers need to be aware of now in order to make this an easier process for them in future? On the other hand, we'll also sort of develop a bit more of a technical overview of the project, which to be confirmed exactly how, but it's probably more suited maybe to a conference setting, or at least in detail in the conference setting. And the focus there will really be, well, what do these findings mean for, policy strategies moving forward, and what's the implication, for the industry when it's confronted by these overarching emission reduction targets that have been set by other stakeholders. And we'll also make those future research recommendations too. So what the outcomes of this are is, as described in sort of our project overview, our key stakeholders will have access to this publicly available information, particularly around, the cost and the level of ambition and adoption needed to achieve these emission reduction targets. At the same time, doing this at a whole of industry level will also provide some core assumptions and methods for those companies as they do move forward and have to set their own emission reduction targets. We're kind of providing a yardstick of, well, this is what's required for the whole of industry. This is how you might go about doing it. And we're also hoping to support the credibility of industry's commitment to environmental improvement. That's a long demonstrated commitment, including in the space of, greenhouse gas emissions. The chicken meat industry was one of the first to do sort of a carbon footprint LCA type assessment and publish that. And there's an ongoing portfolio of work in relation to not just greenhouse gas emissions, but the broader environmental portfolio. However, to bring it back to what we said at the start, our outcome here is really to support industry and all stakeholders across industry in relation to these, external targets that have basically said you must achieve this, and left it completely up in the air as to how that's going to be implemented and what the cost is, and really whether it's actually technically feasible at all. So just to wrap up, thank you to AgriFutures for funding and supporting this project, and also to the members of our project steering committee and other industry stakeholders who supported and contributed to this project. Very much look forward to sharing the results with you in due course.
Fran, thanks very much for the presentation. And as you sort of mentioned in some of those closing slides, we'd certainly love and plan to have you back on the series to provide the final sort of outcomes to the broader industry. So I will move over to introducing our last speaker for this webinar. So Professor Dana Stanley has a PhD in molecular microbiology from Victoria University, and after receiving her PhD, she spent time at CSIRO Australian Animal Health Laboratory, now the ACDP, and specialised in gut microbiota and genetics during her time, and this was under the supervision of Professor Rob Moore. She moved to Central Queensland University and is now the leader of the Molecular Microbiology and Poultry Health Research Cluster in the Institute for Future Farming Systems, and focuses on human and livestock intestinal health and nutrition, probiotics, next generation antibiotics development, and pathogen control. And interestingly, several of Dana's projects are running directly on farms under full production conditions. So during this last part of our webinar, we'll hear from Dana about her project, Integrated Investigation of Enterococcus cecorum. Dana, over to you. Thank you.
Thank you, Sarika, for introduction. I'm really happy to present the project funded by AgriFutures, Integrated Investigation of Enterococcus cecorum. And Enterococcus cecorum, I believe everybody in the audience today knows about Enterococcus cecorum as a major global emerging broiler pathogen and becoming a really big issue in veterinary medicine. It is hard to keep up with all these new pathogens coming. But Enterococcus cecorum is certainly in fashion nowadays. Enterococcus infection is primarily affecting skeletal system, leads to lameness and paralysis. And we have lesions in the heart. We often have pericarditis, and then we also have infections in liver, like hepatitis, and then also femoral head osteomyelitis. It's characterized also by free thoracic vertebra infections. So the birds are suffering immensely, and it is not very pleasant thing to see in the shed or to watch birds suffer that way. It's often preceded by intestinal colonization, it is often preceded by intestinal colonization, but it's often done with different strain. Antimicrobial resistance and increased mortality, decreased feed efficiency, poor meat quality, cost of veterinary treatments, and of course, problems with reputation of farmers if someone turns up and sees animals birds in this condition. We applied for this project based on preliminary data that we gathered by doing online sort of analysis, because every manuscript that uses 16S or any type of shotgun sequencing data has to submit all of their data to public databases before they're allowed to publish. We used this abundance of data on public databases and downloaded all 16S microbiota studies ever submitted. Well, that was 2 years ago. And separately, all shotgun metagenomic studies that are done in poultry. Out of 1,503 manuscripts that passed all of our rigorous sort of filtering, we ended up with doing analysis on 34,838 FASTQ files. These are paired sequencing files. So this is the number of samples that we analyzed, and they were fully annotated, only fully annotated for the breed and all other sort of interesting sort of information. We also did shotgun metagenomics, manually annotated about 4,000 metagenomes and analyzed that. Annotation was done by reading materials and methods, so we have exact knowledge of what was done to these birds, what they fed, and all of other information. 20 million CPU hours and 500,000 jobs submitted to HPC, and we ended up getting a massive data set. And then we had a look into Enterococcus. We found these species of Enterococcus here using shotgun metagenomics data, and we found few interesting things. We found that it's more prevalent in breeders than in broilers and in production, and then that it's also a bit more prevalent in male than in female birds. We saw that it's highest in esophagus. That can indicate origin from sand or dust or inhalation, or ingestion. We also noticed that it's very high, the peaks across all of these data sets. It peaks on day 0, so it's probably there from hatchery or from beginning or from the breeding stock. Those were our hypotheses that we brought with us when we applied for this project. We also saw that there is significant difference using either 16S or shotgun when we look into the breeds. So there is a different susceptibility. And it's very interesting finding for us here was that we need to look into day 1, into hatcheries, into broilers, as well as into breeders. So we also have, my team actually, we have lots of experience with building networks. So more data you have, more reliable are your co-expression, or in this case, co-abundance networks. So what we're seeing here, this is everything significant out of these 40,000 samples that we have analyzed. These are only significant interactions between genera. We're seeing Enterococcus here in a hub, being positively correlated with Staphylococcus, Gallibacterium, Streptococcus. So we are, and Clostridium, the red lines are positive correlations. That means that across all these data sets, they go together up, they go together when they go down, which means that there is strong connection between them, that kind of in plain language that they're working together very often. Interesting here is this link, positive correlation with lactobacillus, something that we want to look into as well during our project. And we have a lot of experience with networks, and a lot of networks published already across different molecules. We were lucky to get Enterococcus outbreak samples, and our preliminary data showed that indeed we do have co-infection. And co-infection is something that is becoming very popular in human research as well, because we are abandoning that simplistic approach that there is one pathogen, one disease. There is lots, it's much more complicated than that. In this case, birds affected by, like from the disease outbreaks, actually the green line here is Streptococcus pneumoniae. We can see bone marrow, only shotgun sequencing detected only this strain. And then we are seeing other bone marrow samples all having Streptococcus pneumonia, liver having Streptococcus pneumonia, and 3 bones having Enterococcus also. So we were confident that our network analysis and that our idea that this is much more complex is going to be confirmed in future experiments. Our main objectives are looking into genetic diversity. So we are already, I think we have hundreds of Enterococcus cecorum colonies isolated, typed, and we are now trying to select different isolates from different outbreaks, from different origins, and to look into look into virulence factors, to look into AMR resistance genes, and their relationship between, for example, Victoria, Queensland, and other parts. And we're trying to find some things that connect them in some ways. We're also looking into epidemiology, like where are the origins of this infection, of this of disease outbreak specifically. And then we are also looking, obviously, co-infection, which is one of our priorities. And our objective 4 is targeted mitigation, to come up with some advice for the growers and for breeders on how to control this and how to reduce the incidence and how to try to prevent it if possible. The way we approach this issue is investigating EC prevalence in both and genetic diversity in both breeders and broilers. We are doing strain typing, identifying and characterizing transmission routes. Through shotgun metagenome, we look into interaction networks, and we are also including cell culture. So not just culturing bacterial pathogens, but we are also working on co-culture experiments to identify how they interact inside within the sort of inside the bird. For that, we use some chicken cell lines. Actually, we are using chicken organ, sort of primary organ cultures, where, for example, we clean the brain, fresh ileum, clip the ends, and inject different pathogens into the ileum. We do this like the experiment that my students call ileum sausage. Sort of experiments where we look how they interact inside of the bird, as well as other types of chicken cell lines. We developing targeted mitigation strategies. It's too early for that, but we're working on it. We are also using 16S shotgun and PCR and culturing. For example, in some cases, we don't have enough pathogen in liver we detected with PCR, but we don't have enough to pick it up with shotgun or 16S. Then we culture it on plate. When we figure out which one it is, we select the media, culture it, and then we shotgun and identify. So we are doing all sorts of things. This is our sampling logistics. We work with farmers. We're sending samples. We send them eskies. We bought really good eskies that can last for days with a little bit of ice in there. These are the sort of sampling protocols, laminated sampling diagrams with what goes into which thing, and there's also video accompanying that. We send that to the farmers, and we are getting samples from them. Methodology, as I said, we're using shotgun metagenomics. We are using it on bone section, liver, spleen, lungs, feed, floor, range, water supply, dust on the walls, insects, cockroaches. We're looking for mouse pellets. We want to map pathogens, not just Enterococcus cecorum, because while we're doing this data, we will pick up everything else that can be found there, including viruses, including all other sort of types of pathogens. So strain typing, epidemiological profile for each shed separately, and then comparing sheds. We're looking for triggers in metadata. That depends on metadata collected for us by farmers. Then whole genome sequencing will be performed on isolates from different outbreaks to confirm genetic diversity and identify more factors. We will also co-culture Enterococcus cecorum with range of these other sort of pathogens with which it co-infects, and using a gut simulator fermentation system to look into how they interact and try to figure out how they talk to each other by looking into metabolite analysis. The outcomes, commercialization roadmap, clear roadmap on hopefully for commercializing some perhaps vaccines or treatments or sort of disseminating our farmers with findings and discussing them and refining them over time through our conversation with farmers. Maybe diagnostic kits for EC detection, understanding how it becomes resistant to antibiotics, and more of the mechanisms. The next stage for the future proposals would be intervention strategies once we have all of this data to look into which molecules can break the quorum sensing and communication between co-infecting species, because they communicate through molecules. And research shows that a lot of phytogens, for example, in plant products, can block the communication between the co-infecting sort of in co-infection. We are also hoping to look into host response and also drug repurposing through machine learning. And hopefully after we finish this project, we will be able to get into this, to integrate all of this data into prediction system and try to figure out how this all happens. So at the moment, we are doing a lot of culturing, media designing media to grow a lot of different species, and we also use aerobic, anaerobic. We are collaborating with experts in the area. Hao Van, who is now running Rob Moore's lab after Rob retired, and Lido Malecki from Pat Blackwell's lab, they sent us a lot of their own strains of Enterococcus cecorum. They helped us quite a bit. They co-supervise our students. We're also doing obviously a lot of sequencing, annotating, histology on all of these organs, getting ready for co-culture. Just quickly before I wrap it up, we are finding a lot of interesting things. We are finding, for example, I'll just share one thing that since there is 15 minutes is really not enough to show any data. But we are finding that oviducts, for example, are infected already with E. coli and Enterococcus, both. We're finding them in oviducts across different farms. We're not finding anything on eggshells, swabs from eggshells, whereas we were 100% sure we will be finding these things in incubators and in eggshells, but we are actually finding it in oviducts. We are looking forward to sharing all of this data. In case there are people here who would have opportunity to send us their outbreak samples, I would like to ask you to please contact Sarika or contact me as well, and we will have a chat about that. We are really interested in outbreaks across Australia. Also, all outbreaks are of interest, because Enterococcus cecorum can play a role in them. And if you send us some samples that are not of Enterococcus cecorum, once we determine that it's not Enterococcus cecorum relevant, you will still get a lot of PCR data. We're just not going to proceed into the sequencing and more expensive parts. We are really interested in how Enterococcus plays a role in other outbreaks as well, because we're really willing to understand how this disease outbreaks happen and what leads onto them. So this is the team that we assembled at CQU. Giselle is our lab tech. We have Emmanuel Asara. He's one of our PhD students. Sung Yu Leo is postdoc. And that's me and Yadav. We are two chief investigators. This is our HPC support, because we're doing a lot of AI computing and prediction modeling. We need really good IT specialists, and that's Jason. And Rod Jenner is missing, and Rod Jenner is a ghost person, very hard to find. But he's still actively involved in the project and gives us really precious information and advice. The second student that is going to start pretty much in a few weeks, his enrollment is nearing the final stages, is David, who actually, surprise, surprise, did his master's on Enterococcus cecorum comparative genomics of broiler chickens with Soy Rubright, Peter Scott, and Hao Van. So this is the benefit of having friends in good labs. This is it from me. I also want to thank, I'm really grateful to AgriFutures for allowing us to explore this project together, and for being so professional and easy and good to work with. Thank you very much.
Thank you very much, Dan, and thanks very much for the kind words at the end. What I'll ask you to do, I'll ask you to keep your video on, but I'll ask you to stop sharing your screen. And I'll invite our other presenters to turn on their videos, so that we can have a short Q&A for the remaining sort of 9 minutes that we've got here on this webinar, just so that I can conclude and let you all out on time. So I'll start with John and Sam. I'll start with you guys first. There was an interesting question that was put in, and from one of our audience members. So as AI, so artificial intelligence rather than avian influenza in this context, and automation is likely to sort of sweep across the agricultural industry, and likely, or could it replace behavioural science by programming and robots? They've sort of acknowledged that it might be a bit provocative, but very interested on your thoughts and thinking in this space.
Thank you, Sarika. I'm happy to give this one a crack, John. And thank you for the question. It's certainly not provocative, a great question. Maybe the best way to answer this, I mean, we're really firstly just fascinated about the role of AI and where it's all going. I think even the last year or two, just seeing what's happening in that space, it really is growing and expanding exponentially. So it is definitely a fascinating space to keep across. But yeah, look, I think the best way to probably answer this one is to go back to John's initial introduction about our role and the type of work we do in the project. And I think generally from our view is if there's anything that's involving, people making some kind of decision or being impacted or interacting, going through some kind of transition, whatever it might be, we think there'll be an important role for behavioral science, because we need to understand, what that looks like, what types of groups are impacted, and how do we get messaging and programs out there to hopefully help with that transition or get more engagement, whatever it might be. So that's a very quick response, but it's a fascinating topic nonetheless.
John, anything further to add to Sam's response there?
Oh, I was just thinking, Sarika, that Evidn, we work here in Australia and also in America. I'm not sure if I'm more worried about AI or what's happening on the human front. If you think about agriculture and policies and things that are happening at the moment in real time, so we'll see. There's a lot to be seen there, but I think there's always. I sometimes describe Evidn like an analog in a digital world, and I don't mean that in a negative sense of the term, but I think where the skills of behavioral science come into play is when humans are obviously involved in the decision making, and I think there'll always be some role for that. Maybe a different looking one, but perhaps some. So there you go.
Certainly, thank you very much, and I think that the point that you made about the impact of policy on agriculture is, and it's sort of a nice segue to some of that content that Fran had presented. And Fran, we did have a couple of questions that came through in response to your to the slides that you'd shared. So the first is around sort of cooperation, and the question was: is the project building cooperation across the industry's value chain? And the asker of the question noted that a lack of cooperation seems to be a barrier to reducing emissions.
Yeah, it's a great question. Look, there's kind of, I think, two components to that. I mean, first, like at the project team, we've been quite pleasantly surprised by how, all the stakeholders we've engaged with via the steering committee, how, open and supportive they have been to, refining assumptions and basically contributing to the project and wanting a good outcome. With that sort of goes hand in hand, you are getting an insight and some solid, quantified numbers into where, emissions differentiate across stakeholders and across the supply chain, but it's also going to provide insight into, well, where does the greatest potential lie and how does the cost for emission reduction differ, for a contract grower versus a major vertically integrated processing company versus, a mid-tier processing company. And I think all of that is going to support, those conversations moving forward. We are in a bit of an unknown state in that, we haven't been able to put numbers or timeframes really around these things, but, at all levels being able to say, well, this is what our profile is, this is what's viable here, will help that contract grower, will help that integrated company, engage with their superior, whether it is that company or whether it is that top tier customer or government, around just what is practicable for the chicken meat industry whilst it continues to be our most consumed meat protein.
I think, all of our projects, the researchers who lead those projects, they do engage with as wide of the value chain as they're able to, but particularly the last two projects, the project that Evidn are running and this one, there's been a lot of fantastic engagement to date from a number of representatives across the value chain. There was a second question that was in the chat regarding your presentation. I think you did answer it when you sort of covered the methodology, but more just a bit of a clarification around, it was a query around how you're obtaining the required data for the project. Have you found with the approach that you've taken that it's lent itself to allowing you to collect the required information to achieve the project outcomes?
Yeah, definitely. So, I mean, we have very good data sets as it is for sort of baseline data, and some costs are quite readily available in terms of, well, what does a 10 kilowatt solar system cost? But it's, I guess those more sort of intangible or variable things, like what would it cost to reduce crude protein by 5% on an average across the industry? Like, we're not the best to answer that. And so that's where, that engagement with the stakeholders from each company and each section of the supply chain has been really valuable. People have readily acknowledged, this isn't my area, I suggest you go and ask this person. And that's, helped, I think, give a really high quality and high confidence in the data, and hopefully the results moving forward.
Yeah, that's wonderful to hear, and I'm sure, certainly the program team, and I'm sure our audience in the wider industry will be interested to hear about that when it is completed. And finally, Dana, a question come in for you, and it was a query around whether Enterococcus in breeders, whether it's more prevalent in younger or older breeders.
So I could not answer that based on just one sort of breeding farm sample. I'm going to use this opportunity for shameless project promotion and just ask for samples. What we intend to do with each batch of samples that we get, hopefully samples with really good annotation, is to, first thing we do PCR on all possible pathogens. Then we do 16S on all samples, no matter how many hundred you send to us. And then we select those that we're going to do shotgun, deep shotgun, and then also culture the strains. So I hope that industry can see, the farmers can see the value of sending the samples to us, and information they're going to get is going to plot not just Enterococcus cecorum, but all of the pathogens. More farms we have, stronger the data, better p-values, better, more reliable answers, and more reliable sort of hopefully mitigation strategies should come out of this.
Fantastic, thank you, Dana. And perhaps this is one that we might revisit when the project is sort of further in, when we have another webinar and get a bit more of an update on how the project's going at that stage. So we've hit sort of 3 o'clock on the dot. Just very quickly before our audience leaves, I'd just like to, in closing, bring your attention to the post-webinar survey that will show up on the screen. We do greatly appreciate your time in providing feedback so we can make any improvements to the upcoming webinars as part of the series, or where feasible, to try and feature some of the suggested topics that are provided. Following this poll, there'll be another poll that comes up, and that's just for us to sense check the platform that we're hosting the webinar on, which is Zoom, and just understand if that's working best for our audience. As a quick reminder, we will provide all of the links to any resources mentioned during this webinar, and where the recording will be housed, which is on the AgriFutures Chicken Meat extension page. If you did register for the webinar, you'll get that automatic email a few days following today, just giving you a link directly where to go. And as always, it's just a reminder to please keep your eye out and report anything unusual or concerns about a potential emergency animal disease to the EAD hotline. And finally, and last but not least, I'd like to extend a really big thank you to our presenters, so Sam, John, Dana, and Fran. We appreciate your time today to share the research that you have underway, and really look forward to working with you to deliver those anticipated and exciting outcomes over the course of the projects. And of course, thank you very much to our audience that have registered and or joined. We'd like to wish you a fantastic rest of your Tuesday, and we'll see you next time. Bye for now.